So we actually study population genetics by looking at allele frequencies. Now you can't necessarily look at an individual and always know what alleles they carry, but you can look at some individuals and figure it out, especially when you know something about the gene in question. When populations evolve, their allele frequencies change. And evolution is actually defined by change in allele frequency over time. So this sort of grid is how our book represents allele frequency. So we're going to see this quite a few times in this chapter. They're just giving you a visual display of allele frequencies. So they might also refer to these as haplotypes. With each circle representing either a particular haplotype or a particular allele if they're talking about a specific gene. So you would study a population by counting directly or computing the frequency of each allele in the population. And you can do this using a variety of techniques, gamete sampling, looking at if you can harvest gametes from the individuals. You can do genetic analysis using microarrays or DNA sequencing, or you can actually just observe the population if you're interested in a particular trait that has a visible phenotype that you can recognize. So from this grid, we have the blue and yellow alleles, and they are calculating a frequency by just counting up the number of each type of allele and dividing it by the total number of alleles in a population. So these are representing alleles, not individuals in this case. So every individual has 2 copies of each allele, so this would represent 50 individuals. So in this particular example, the frequency of the blue allele is 70 out of 100, or 0.7, and the frequency of the yellow allele is 30 out of 100 total alleles, or 0.3. So we calculate a frequency, whether it is an allele frequency or a genotype frequency or a phenotype frequency. Whatever frequency you are calculating is going to be the number in your subset divided by the total number of whatever you're counting. So if we're looking at an allele frequency, it's going to be the number of a specific allele occurring, such as the blue or the yellow in the previous slide, divided by the total number of alleles for a gene. If we are looking at a genotype frequency, it would be the number of individuals with a specific genotype divided by the total number of individuals. So when we're doing this chapter, keep in mind what you are actually studying. If you're looking at genotypes or if you're looking at alleles. Remember that if you are calculating allele frequencies, each individual has 2 alleles for any given gene. We can measure change in a population by looking at how allele frequencies change. So we're going to start out with the idea that the frequency of all alleles together in a population is equal to 1. Therefore, the population is made up of the sum of all of the allele frequencies for different alleles. So by convention, we are going to use the lowercase letter p to represent the frequency of the dominant allele, and q to represent the frequency of the recessive allele. It does not have to be dominant and recessive. Sometimes you don't know which one is which, and that's okay. For our problems and for our class, I'm going to say use P for the dominant allele and Q for recessive. And I'm going to talk about the equation that way. But this makes sense because if you have 70 out of 100 alleles equal to P, that's equal to 0.70. And your Q alleles are 30 out of 100. That's equal to 0.3. If we add P plus Q, we get 100 because the 70 and the 30 are a subset of the total. So when you add them up, you can only get the total. So our total is going to be 100 out of 100, or 1. So P plus Q equals 1. It always equals 1. It never equals more than 1. Therefore, if you know 1, you know the other. You can calculate it. A genotype frequency is just going to be the product of the individual allele frequencies. So if we have an individual who is homozygous dominant for the homozygous dominant, and let's say that P represents the A allele, and our frequency of the A allele is 0.7. The frequency of that homozygous dominant situation is just going to be the frequency of one allele in that individual, or sorry, in that population, times the frequency of the other allele in the population. So it doesn't actually matter what's here, we're just multiplying the alleles. So the frequency of homozygous dominant individuals is going to be 0.49. Now this is the same as saying the frequency of the homozygous dominant is equal to P squared, or 0.49. So the frequency of the homozygous dominant is 0.49. We can do the same thing if we want to figure out the homozygous recessive. So in this case, the frequency of homozygous recessive is just the frequency of the Q frequency, the frequency of Q, 0.3 times 0.3, which is going to be 0.09. Now that is the same as saying Q squared. So Q squared is our frequency of homozygous recessive and is equal to 0.9. So this means that 42% of the population is homozygous dominant and 9% of the population is homozygous recessive. Now we can do the same thing for the heterozygous individual. So the frequency of the heterozygous individual is going to be the frequency of the P allele times the frequency of the Q allele. But we want to point something out. This big A, little a situation can come from either mom giving the dominant allele and dad giving the recessive, which would be 0.7 times 0.3, or mom can give the recessive allele and dad can give the dominant allele. The chance of that is 0.7 times 0.3. But because there are two ways, we actually have to use the sum rule. There's two ways that an individual can be heterozygous. So we actually have to say that the frequency of the heterozygous individual is equal to 2 times 0.7 times 0.3. So the frequency of heterozygotes is the same as 2 times P times Q. In this case, that is 0.42. So this means that 42% of the population is heterozygous. So remember that these are all frequencies. A frequency is going to add up to 1. So if we take all of the individuals in a population, P squared plus 2PQ plus Q squared, that accounts for everybody, both the homozygous and the heterozygous individuals. We have 0.49, 49% of the population is homozygous, dominant. 0.42 or 42% of the population is heterozygous, and 0.9 or 9% of the population is homozygous recessive. If we add that all up, we get 1. So we've represented everybody in this population. We have also come to the Hardy Weinberg equation. So the Hardy Weinberg equation helps us to calculate genotype frequencies. So those genotype frequencies are based off of the allele frequencies. Now, if your allele frequencies change, evolution has occurred. There's been a change in your population. And in most cases, well, in all cases, if your allele frequencies changes, your genotype frequency has to change as well. So based on the previous slide, previous discussion, the P squared part of our equation is the expected frequency of the homozygous dominant genotype. The 2PQ is your expected frequency of the heterozygous genotype, and your Q squared is the expected frequency of the homozygous genotype. Now, if an individual has the homozygous recessive genotype, you can use that to calculate your Q value, or your homozygous allele frequency, your recessive allele frequency. So all individuals that are homozygous recessive, you know their genotype. If you have individuals who are showing the dominant phenotype, they have either the homozygous or the heterozygous genotype, and you can't distinguish them. So you cannot calculate P directly from your dominant individuals because you don't know how many of those individuals are actually hiding some recessive alleles. You can, however, calculate Q from your homozygous recessive genotype frequency. So here's an example that shows how we might use our allele frequencies to calculate our genotype frequencies, and it's just another representation of how we get our P squared and 2PQ. So this diagram represents it as a Punnett square. We've got flowers here. We're actually looking at an incomplete dominance situation. So our homozygous individuals are red or white, and our heterozygous individuals are pink. I like this because you can actually in this situation see the difference between the heterozygous and homozygous individuals. But I also like that it demonstrates that with your 2PQ, it shows your reason for the 2PQ. There are two different genotype combinations that give you the pink individual. So P squared is going to be the frequency of the red phenotype is equal to the product of each of the allele sequence frequencies. The white phenotype frequency is going to be equal to the product of the recessive alleles. So here's our book's representation of this, this equation. If you have a population where the blue allele is occurring in 70% of the alleles are blue, 30% are yellow, and then you allow those individuals to mate, and for random fertilization to occur, you just end up with this distribution of genotypes, and represented within that distribution of genotypes are our different phenotypes. We've got our heterozygous and our homozygous individuals. You can see those represented here: heterozygous individuals, homozygous individuals. You can see again that this is just the product of our allele frequencies, because this is just a chance event. It is the chance that any individual is going to get either version of the gene. So if we wanted to use this to determine whether or not a population is changing, we would look at the genotypes, particularly the homozygous recessive genotypes. At one point in time, we could calculate the allele frequencies and then do the same thing and look at the population again after some sort of event that might cause a change. We'll talk about those in a few minutes, and look to see if the allele frequency has changed. We can also use Hardy Weinberg to get some information about our population. So here's an example problem using RH factor. Remember, RH is one of our blood typing factors. So 16% of the global population is homozygous recessive for Rh negative blood type. When you are given a Hardy Weinberg problem, the first thing you need to figure out, very first thing you need to do, is determine whether or not you have been given information about allele frequencies or genotype frequencies. This is the number one mistake that students make on these problems, is determining whether or not you are being given information about P or Q, or whether or not you're given information about Q squared. So I told you that 16% of the population, if we write that as a frequency, it's going to be 0.16. 16% of the population is homozygous recessive for Rh negative blood. I want you to pause for a second and think about what I gave you. Did I give you an allele frequency or a genotype frequency? If you are given an allele frequency, it's probably going to say something like the allele frequency of the Rh negative of the Rh negative allele is X. Or it's going to say something like, in a population of 200 people, 14 have the Rh negative, 14 individuals are Rh negative. That one you could go either way, but you have to read the problem carefully. In this case, you are being given a phenotype. You are being told that individuals are Rh negative. This is an observable phenotype. So if we go back to our Hardy Weinberg equation, when we are given the homozygous recessive phenotype, we are given the Q squared part of our equation. So if we are given the Q squared, we are given a frequency of Rh negative individuals. So we're told that Q squared equals 0.16. Now we can determine the frequency of the Rh negative allele. And we do that by simply taking the square root of our homozygous recessive phenotype frequency. So we know that the frequency of that Rh negative allele is going to be 0.4. Once we know the frequency of that recessive allele, we can use P plus Q equals 1 to determine the frequency of the Rh positive allele. So P is going to be equal to 1 minus Q. P is equal to 0.6. So now we know the frequency of the Rh positive allele and the Rh negative allele. Oh, I didn't even need to erase that. All right, so let's figure out what is the frequency of homozygous Rh positive. So we know from our calculations that Q is 0.4, P is 0.6. Individuals who are homozygous for Rh negative, we'll call them big R. That's just the frequency of the big R allele times the frequency of the big R allele. So this is going to be P squared. So P squared is 0.6 squared. 0.6 squared is 0.36. So this means that 36% of the population are Rh positive for their blood type. We can also figure out the frequency of the heterozygous individuals. These are the individuals that are going to be big R, little r. Remember, that can happen in two ways. That can be big R, little r, or little r, big R. So 2 times P times Q, 2 times 0.6 times 0.4 is equal to 0.48. Now we can check this and make sure that our P squared plus 2PQ plus Q squared is equal to 1. 0.16 plus 0.48 plus 0.36. I'm sorry, I just wrote P squared and Q squared backwards. But we can check to make sure that this is all equal to 1, and it sure is. So now we have some information about our population. We know that 48% of our individuals in our population are carriers for Rh negative, and we know the composition. So now we can, if we wanted to see if the population was changing at another point in time, we could determine the frequency of our homozygous Rh negative individuals and see if our allele frequency has changed. So sometimes our allele frequencies and genotype frequencies do not change. If allele frequencies stay the same, it means that the population is not evolving. If allele frequencies stay the same and random mating is occurring, then our genotype frequencies are not going to change, except they might fluctuate a little bit just by chance. But your allele frequencies are going to stay the same. So this diagram is just showing that if these individuals on the left have a next generation of offspring, about half of the alleles are blue, 42% of the individuals are heterozygous, half of the alleles are blue, half are yellow. Our frequencies are, and then I missed the bottom, 9% of our individuals are yellow. They contribute all yellow. So if we use that information to calculate the next generation, we see that nothing has changed. Our P and our Q have stayed the same. So the converse of what I said at the beginning is that if no evolution is occurring, then allele frequencies and gene frequencies stay the same. So if there's not some sort of outside force that is causing those allele frequencies to change, then evolution is not occurring. So there are certain evolutionary forces that will cause the allele frequencies to change. Remember that I said that evolution occurs when alleles change, so our allele frequencies change. So our measurement of change in a population is going to be those allele frequencies. So we use the Hardy Weinberg equation to describe a population that is not evolving. We say that a population that is not evolving is in Hardy Weinberg equilibrium. And in order to say a population is not evolving, we have to make some assumptions. There are 5 Hardy Weinberg assumptions. They are random mating, no mutation, no migration, extremely large population size, and no differential reproduction. No differential reproduction is another way to say no natural selection. So if a population is in equilibrium, none of these things are occurring. The flip side of that is that if any one of these things changes, if the Hardy Weinberg assumptions are violated, then we are going to have a change in the structure of the alleles in the population, and that evolutionary change is occurring. So evolution occurs if any of these are violated. So this table just shows what the violation is. They're all pretty straightforward. If we have non-random mating or assortative mating, we'll talk about what assortative mating is in a few minutes. If that happens, we actually only get a change in genotype frequencies. So allele frequencies don't actually change when we have non-random mating. So random mating is the only situation where our allele frequencies stay the same, even though our genotype frequencies change. For the other 4, if migration occurs, if individuals move in or out of a population, our allele frequencies change because individuals come in or individuals go out. If mutation occurs, we now have a new allele, or a mutation may also occur changing one allele into another in a particular population. If either one of those occurs, we're going to change allele frequency. So a new allele might come about, in which case that by default sort of decreases the frequency of the other alleles. Or you might have forward and back mutations, converting one allele into another. Either way, that's going to change your allele frequencies. If we do not have a large population size, genetic drift or chance changes in allele frequencies, chance fluctuation in allele frequencies are going to occur. And then, of course, natural selection. If you have a trait that is being selected against, it's going to probably be removed from the population, and so the frequency is going to go down. If it's an asset to the individuals, it's going to be passed on more frequently, and it's going to increase in frequency. So you do need to know all of these. You need to know the assumptions, so that for Hardy Weinberg equilibrium, you need to know the way that they are violated and how it impacts the allele frequencies in a population.